A practical workflow for making anatomical atlases in biological research. Biologists, artists, and computer scientists at the University of Utah work together to make a mouse limb atlas for studying muscle patterning. The workflow is adapted from a CG artist pipeline and is usually for building 3D models for animated films and video games. In this video, we present the workflow and the resulting atlas. Data acquisition. We use confocal laser scanning microscopy to capture 3 channels of different structures of a 14-day embryonic mouse. As shown in the volume rendered result, the data contains muscles, tendons, and nerves of the mouse hind limb. Segmentation. We use Photoshop to segment the muscles from each image slice of the volumetric data. We first adjust the image brightness and contrast with the Levels tool. Then we use the Quick Selection tool to paint the muscle to select it. We use fluorender to extract complex structures such as nerves. With fluorender's selection brush, we paint directly on the volume rendered result and select the desired structures. The selections can then be extracted and saved as a stack of images. Modeling. We use Maya to make the polygon models for different structures. For spindle-shaped muscles and tendons, we first import the segmented data into Maya, and then load a prototype model. The prototype model is reshaped to match the segmented data. For other regular-shaped structures, we create a box and reshape the box to match the shapes of the segmented data. For branching structures such as the nerves, we start building the model from one end of the structure and extrude the faces. Texturing. For detailed structures such as the muscle fibers, we paint textures on the polygon models with Photoshop and Mudbox. We first load a volume rendering of a muscle into Photoshop. We then paint muscle patterns with Photoshop's brushes. The painted pattern is used as a stencil in Mudbox. We import the muscle model into Mudbox and then load the stencil image. We can adjust the stencil image for projection. The muscle patterns are projected onto the model by painting with Mudbox's projection brush. Results. The results are high-quality textured polygon models for the limb atlas. Users can interact with the models easily. Models can also be colored differently. Users can adjust the transparency of the models. Underlying structures can then be revealed. Evaluation. We made a total of 122 models for the mouse limb atlas, including 59 spindle-shaped models, 36 tubular models, 10 spherical models, 7 branching models, 6 flat models, and 4 irregular-shaped models. The 3D pie chart shows the average time of making one model for the 6 shapes and 4 processing steps. The total time for completing the atlas is about 120 hours. We believe our workflow is efficient for making high-quality anatomical atlases in biological research.